Ever wondered how scientists grow bacteria, fungi, and other tiny organisms safely and effectively? Welcome to today's video, where we'll go into microbiological incubators and the role of the BOD, Biochemical Oxygen Demand incubator. By the end, you'll know how these devices work, what makes them unique, and the step-by-step process for incubating microorganisms successfully. In laboratories, incubators are vital tools for growing microorganisms. Whether you're a researcher, a student, or just curious, understanding the purpose and function of these devices is key to studying microbiology. Let's break down the basics of microbiological and BOD incubators. A microbiological incubator, often called a standard incubator, is designed to create the perfect conditions for microbial growth. This device controls temperature, humidity, and sometimes CO2 levels, essential for keeping microorganisms happy and healthy. It's widely used in research labs for culturing bacteria, yeast, fungi, and even cell cultures. Typically, a microbiological incubator maintains temperatures between 20 degrees and 45 degrees Celsius. For most bacteria, a temperature around 37 degrees Celsius is optimal because it's close to the human body temperature, which many pathogens prefer. Now let's talk about a special type of incubator, the BOD incubator. BOD stands for Biochemical Oxygen Demand, a term often used in environmental sciences. BOD incubators are built for applications that require precise, low-temperature settings, ideal for growing microorganisms that thrive in cooler conditions or studying environmental samples. BOD incubators can maintain temperatures as low as 5 degrees Celsius and up to 60 degrees Celsius. This feature makes them perfect for water testing, soil microbiology, and studying metabolic rates. Unlike standard incubators, which are generally warmer, BOD incubators are often used for long-term experiments where stable, cool temperatures are critical. So, what makes these incubators so precise? Both types have advanced digital controllers that allow researchers to set specific temperatures. Many models also come with alarms to signal any temperature fluctuations, preventing costly errors. Additionally, some incubators have double-walled doors and airtight seals to maintain a stable internal environment. For added flexibility, incubators have adjustable shelves to fit various culture dishes, petri plates, or flasks, making them versatile for different experimental setups. Now that we know the difference between a standard microbiological incubator and a BOD incubator, let's go over the steps for incubating microorganisms. First, gather your sterilized petri dishes or culture media. Label each plate to avoid confusion later on. Using a sterilized inoculation loop or pipette, transfer a small amount of your microbial sample onto the media. Make sure to work under sterile conditions, such as in a laminar flow hood, to avoid contamination. Choose the appropriate incubator based on your sample's needs. For most bacteria, a standard microbiological incubator set at 37 degrees Celsius works well. For soil and water samples, a BOD incubator set between 20 degrees Celsius and 30 degrees Celsius may be more suitable. Arrange your samples inside the incubator, leaving enough space between plates for air circulation. Close the door tightly and let the incubator work its magic. Check on your samples periodically. Depending on the microorganism, incubation may take anywhere from a few hours to several days. Be patient and avoid opening the incubator frequently, as it can disrupt the internal environment. Incubating microorganisms under controlled conditions is essential for accurate results in experiments and tests. Whether you're studying pathogens, testing environmental samples, or examining growth patterns, the incubator's role in microbiology can't be overstated. And that's it. You now know the basics of microbiological and BOD incubators and the steps to incubate microorganisms successfully. Remember, setting up your incubator correctly ensures reliable, reproducible results. Thanks for joining us today. If you found this video helpful, hit that like button and subscribe for more lab insights. Until next time, keep experimenting and stay curious.